I had a question. You're muted.
Okay, what's up?
Okay, so on this on question 8, is his finger on
Let me
the
go
ball?
check it. Let me go look at it real quick. Let's see. Okay, I'll message you in the chat.
Okay. So I finished and it gave me on number 7, it gave me partial credit because I put
Wait, don't say it. Don't say
I'm
it.
gonna say I put it, but I literally put it the way that it's giving me, but it gave me partial credit still.
Yeah, so I have to manually go in and grade those, so don't worry about your current grade that it gives you. I have to go and manually like check spelling and all of that. So if it gave you partial credit and you're concerned about it, I'll let y'all know what we're gonna do about that issue after the quiz.
Okay.
Alrighty, nice. So it looks like everyone has finished. I think that's what Canvas is showing me. So we'll go ahead and begin today's lab. And if you're still working, which I don't think anyone is, it's not showing me that anyone is. So if you're still working, just ignore me, but if not, we will begin. So I wanted to make a few announcements before lab began. First of all, I got all the grading done except for the last quiz, so the quiz from last week. I think was quiz 3, but all the lab reports and quizzes have been graded. I'm going to ask that if you saw that I had an error in your grading, which most likely I didn't, but I have 45 students' papers to grade, and sometimes it gets really complicated, and Canvas, there's a submission issue that I'm about to go over. So if there was an error, please chat me or during this Zoom at some point privately, and I'll go and take care of that after lab. But what has happened is whenever you submit your lab report, it needs to be in one Word document. If you submit each image individually, it takes so much time for me to load each one. I have to click every single image, wait for it to load, it buffers, and I have to go through each one. So what I ask is that you put it in one Word document with your outline, so you can type up your outline. You take a picture of your drawing or activity or whatever it is and just insert that into the Word document. Then I can just scroll through one thing and grade it a lot quicker. Whenever you do a bunch of different submissions, it takes so long to grade. It's very time consuming. It's super frustrating, especially if you live at the Vistas and the Wi-Fi sucks. So please make sure you put your submissions in just one Word document from now on out. That would be freaking awesome. Other than that, not many more announcements. Our practical, I think we still have 2 or 3 weeks, so we still have some material to go over. Today's lab will be relatively quick, but next week's lab, be prepared for like a longer lab, so have like, you know, a snack to get you through or whatever. These labs technically are 3 hours long. I'm a cool IA and decided to cut them short because who wants to be on for 3 hours? Next week, we might hit like 2.5 hours, especially if the practical is the following week because we'll play a review game, but we'll play it by ear. So I'm gonna go ahead and share my screen. Let me see, where is Canvas? Hopefully the quiz didn't go too bad. It's pretty basic in the world of axial and appendicular skeleton. So let's see. Alrighty, so here is today's module. This is already week 6 of lab. We're on lab 5 because we had that 1 week off for Labor Day. And today we're going to look at joints and body movements, which luckily we looked at body movements the very first week of lab. So that'll kind of just be a review of like abduction, adduction, flexion, extension, things like that. But then we're going to get into muscle histology and anatomy. So we're going to be looking at the actual muscle cells, the differentiating characteristics, and how a muscle cell actually functions. Next week, we'll be looking at where the muscles are located, so like the biceps, brachii, the triceps, you know, all of that. This week, we're just looking at the histology, or the study of the tissue. So this is chapter 9 and 10. We just took quiz 4, and now we're going to start lab 5. We have 2 PowerPoints today. I did make them fill in the blanks, so if you want to go ahead and pull them up, you can fill in the important topics as we go along. But make sure you're still listening because there will be some things on the PowerPoints that aren't blank that I still want you to know. So if I, you know, state like this isn't a blank, but like you still want to know it, make sure you're listening to get that marked down. After we go through these 2, I'll explain the lab report and then we will be done for today. So let's begin. So starting, where is it? Here we go. Starting with chapter 9, joints and body movements. Hopefully you all have this pulled up or printed out or something to go along with me. So our objectives for today will be looking at the joints and classifying them according to their structural and functional classifications. We're going to look at the structure of a synovial joint, We'll look at the 6 categories of synovial joints and examples of each, and then we'll demonstrate types of movements, which again, we've already learned that, but it'll be a nice review. So, joints. Joints are also called articulations. So if you remember when we looked at the axial skeleton, we looked at those 2 bones of the lower skull, the occipital condyles, and I repeated many times that the occipital condyles of the skull articulate with cervical vertebra 1, the atlas. So anytime you hear the word articulate, that means come together and form a joint, or connecting bones to cartilage, or bones to teeth. So like how I said, the occipital condyles articulate with the atlas to provide for head nodding. That's just the same as how the head of the humerus articulates with the glenoid cavity to provide for, movement of the arm. So articulation just means forming a joint or coming together, connecting. So when we look at joints, we can classify them in 2 ways. We can classify them based off of their structure and based off of their function. When we classify them based off their function, that's when we're primarily looking at synovial joints. Structure, we'll look at 4 different types. So let's start with the structural classification. So the 4 structural classifications we have are fibrous joints, cartilaginous joints, bony joints, and synovial joints. These are based off of their structure. So let's look at each individually. So when we're looking at fibrous joints, these are joints that have little or no movement. An example of this would be like the sutures in the skull. When we looked at the skull, if you remember, we have 2 bones on the top of our head. If we were in person, I'd say, what are those bones? But on Zoom, it takes a long time. But those 2 bones are the parietal bones, the 2 bones on the top of our skull, and they are fused together by the sagittal suture. That suture right there, you can see it on this picture. If you remember, whenever we looked at the fetal skull, the fetal or infant skull has those soft spots where the 2 bones have not fused together, but as you get older, those bones fuse and form a suture that connects those 2 bones and provides little to no movement. We want just a little bit of movement for cases like a concussion, hard impact, things like that. You want your brain to be able to move somewhat inside of your skull, but we don't want it to be able to move a lot. So fibrous joints definitely want to know that there is little to no movement of these joints, and an example would be like the sutures of the skull. We also see it at the joint between the manubrium and the sternal body. So if you remember, the sternum is our chest plate bone, the one right here in our chest, and the manubrium is the upper portion of it. So between the upper portion of the sternum, the manubrium, and the body, there is a suture right here that will provide little or no movement, but that's for cases like if you get high impact to the chest, if an airbag hits you, if you get tackled in the chest, we want just the slightest bit of movement, but not a lot. So what I'm concerned with these structural classifications is knowing the movement and the example. I'm not too concerned about the space. So movement and examples are what you're going to want to focus on here. So that was our first one. Now we're on to our second one, cartilaginous joints. The name of these makes sense, cartilage. So the examples we have here are the intervertebral discs and the pubic symphysis. So if you remember when we looked at the vertebral column, in between each of our vertebrae, we had that little soft cushion, the intervertebral disc that would provide, protection for the 2 vertebrae so they don't, crunch into each other and, there's a lot of friction that would occur there. Instead, there's like this little squishy disc in between them to provide cushioning and protection. And then also the pubic symphysis, if you remember when we looked at the hips, the coccyx, the little piece that formed them together, that pubic symphysis, that's another piece of cartilage that actually rips during childbirth. Woo-hoo. So anytime you see a cartilaginous joint, limited movement, these guys can squish, they can kind of stretch. They of course can't, bend and twist and turn and all of that, but they have the ability to kind of squish a little bit, kind of stretch a little bit, so just limited movement. And again, we see these in the intervertebral discs and the pubic symphysis. Third, we have our bony joints. So bony joints have absolutely no movement whatsoever. So for example, we would see these at the joints between the sacral vertebra and the coccyx. If you remember when we looked at the vertebral column, we have our cervical vertebra, our thoracic vertebra, and our lumbar vertebra. And then extending off of that, we have the sacrum and the coccyx. The coccyx was the little pointy tailbone. The sacrum was kind of the in between the hip bone. And if you remember, the sacrum was 5 fused vertebrae and the coccyx was 4. You're definitely not responsible for knowing the 5 and the 4, but those are completely fused together. So if you, kind of impacted the coccyx, the last vertebra of the coccyx, it would just chip off and the body would absorb it. And done. It's not like it has a little bit of movement where it can kind of bend. Nope, it's just going to chip off. So there is no movement with our bony joints, and we can see an example of these in the sacrum and the coccyx, those vertebrae. And then lastly, we have our synovial joints. These are the most common joints in the body, the ones that you would be most familiar with. So these are freely movable joints. So like our shoulder, our elbow, our knee, all of those joints, our jaw, our fingers, pretty much every, our wrist, all of that is a synovial joint. It is freely movable. Our fibrous, very little movement. It's not like you can move these 2 bones in your skull. Our cartilaginous, the vertebral discs, you can't really move those a lot. They kind of just squish. Our bony, can't move them at all. Synovial, totally and freely movable. So again, we would see these in like the shoulder, the elbow, the knee. And what we see with these joints is it is actually a cavity or a space filled with fluid. So there's fluid in between each joint, so it can have that range to move around. It's not like 2 bones are grinding on each other, or there's cartilage involved. It's just 2 bones with fluid, and that fluid will cushion that movement. So I definitely want you to know that synovial joints are a space filled with synovial fluid. Synovial joint, synovial fluid. So those were our structural classifications. So whenever we do these functional classifications, we're looking at only synovial joints because all of the other joints, they don't have any function. There's little to no movement in all of those. But our synovial are freely movable, so they are functional. So from here on out, we're just looking at synovial joints. I remember when I took this lab, I got kind of confused with that part. So the ball and socket, the condyloid, the hinge, the pivot, the planar gliding, and the saddle joints, these are all synovial joints. You might have heard of a few of them. We're not gonna heavily look in depth at the more rare ones, but I assume most of you have heard about ball and socket joints, maybe hinge joints. So we're gonna go ahead and dive into these. Synarthrosis, antiarthrosis, and diarthrosis, I'm not too concerned with you knowing those. You might look at them in lecture, but I'm in my senior level classes of ESS and I've never heard those terms. I've heard fibrous, cartilaginous, synovial, ball and socket, hinge, things like that, but I've never really heard synarthrosis or diarthrosis. So I'm not concerned with you knowing those. They're just there for reference. But let's begin on our functional classifications. So first we have our ball and socket joint. So a lot of you have heard of this, life science in high school, even middle school, this is one of the most basic joint structures that we've all heard of, a ball and a socket. So, a head of a bone will articulate with like a cavity and form, you know, that kind of angular movement, rotation, things like that. So we could find this at like the shoulder joint. You know, when we looked at the appendicular skeleton, we looked at the humerus, the upper bone of the arm. You know, we all know that's the funny bone. And then we looked at the scapula, the shoulder blade, and we saw how the head of the humerus fit into, does anyone remember what the cavity was called of the scapula that formed the shoulder joint? The blank cavity, anyone remember?
Glenoid.
Yes, good job, Colin. So yeah, that glenoid cavity would be an example of the ball and socket joint. So the shoulder joint, or the articulation of the head of the humerus and the glenoid cavity. So at this point, you definitely need to be aware of your appendicular skeleton, your axial skeleton. If you aren't aware with it by now, you might be a little confused on what I'm talking about. So after lab today, you're like, oh gosh, what was she even saying? Go review your skeleton, and this will make a lot more sense. For functional classifications, I'm definitely concerned about examples. Movements, not too concerned about. Features, I'm not too concerned about. So just kind of know your examples. Ball and socket joint, we can also find it at the hip. So like where the head of the femur articulates with the acetabulum, that cavity of the hip joint, that's also a ball and socket joint. It kind of provides for those, you know, angular movements and rotation, you know, coming forward, back, things like that. So that is ball and socket. Now we're on to condyloid. So condyloid, we're gonna kind of skip through because you don't see many super important condyloid joints. These would be like the joints at the knuckles, or the radiocarpal joints. So kind of like right in here and here. They provide for angular movement, so kind of, you know, stuff like that. But I'm not too concerned with condyloid joints, so you probably won't see them. Then we have our hinge joints, and these are joints that will provide for flexion, flexion and extension. So if you remember, flexion is decreasing the angle of a joint, extension is returning it to 180 degrees. Then we have hyperextension, that would be if you could go, you know, further, like if you were double jointed. But flexion and extension, so that is a hinge joint right here at our elbow. So if you think about it like a hinge on a door, open and close, open and close. So same, you know, we can also see a hinge joint at the knee, you know, open and close, open and close. So that any joint that, you know, opens and closes like a door, that's a hinge joint. Same with our finger, you know, open, close, open, close. So yeah, anywhere you would see like that open and close movement with flexion and extension would be a hinge joint. So now we have pivot joints. An example of this would be the radio-ulnar joint. So radio-ulnar joint, you might be like, whoa, wait, what is that? If you remember last week when we looked at the lower arm, we have the radius and we have the ulna, the radio-ulnar joint. And why it is a pivot joint is because as one bone stays still, the other moves around it. So if you've ever watched basketball or been a dancer or anything like that, you probably know that whenever someone pivots, they're leaving one leg planted while the other leg shifts around it. So in basketball, if you're pivoting, one leg is planted while the other one moves around it. Same in the arm. Whenever your ulna's on the pinky side, radius is on the thumb side, remember, because it kind of makes that R shape with the thumb. So you can feel your ulna touch it. And whenever you flip your arm, you can feel the radius rotate around the ulna. The ulna stays still, and you can kind of press and find your radius, and you'll feel it pivot around the ulna. So if this was my ulna, my radius would go like that. So that. So a pivot joint is whenever one bone stays still while the other rotates around it. And again, the example we see here is the radio-ulnar joint. radio-ulnar joint. You have to know that. Now we'll look at planar and gliding. So the movement here would be gliding. And we would see these in the carpals and the tarsals of the ankle, the carpals of the wrist. These bones, as you move your wrist around, kind of just glide around. They don't, pivot or anything like that. They kind of just softly glide around over each other, things like that. So gliding, gliding. And then lastly, we have the saddle joint. So the saddle joint, these are extremely rare. I actually believe 99% sure we only have one saddle joint in the body, which are the thumbs. So this is just a really odd movement. Saddle joints are what makes humans so superior to other animals. We, have the opposable thumbs, so we were able to grasp things and do things like that, which, helped us evolve into what we are today. So the saddle joint, you can't fully circumduct your thumb. Like if you try to do a full circle with it, it kind of catches you down here and then it pops back over. So when you try to do that full circle, you'll hit a point and then it'll kind of pop you over. If you look at that picture, super complicated shape. So, I guess whenever you get here, it hits, that point and then it shifts you over or something. But yeah, saddle joints, you only find those in the thumb. You might find them in the big toe, but I think you just find them in the thumb. So no saddle joint in thumb. So those were our 6 synovial joints based on their function. And then the 4 we had before were the fibrous, little to no movement, cartilaginous, limited movement, squishing, stretching, bony, no movement at all, and synovial, freely movable. Then we had our 6 synovial joints. We had the ball and socket, elbow, shoulder, knee, fingers, anything that can move around freely. We had our hinge joints, the open and close, flexion and extension. We had the planar gliding, our carpals, they kind of just glide around. Saddle, our thumbs, and then I forgot to condyloid, knuckles, but I'm not going to quiz you on that. And then maybe that's it. So lastly, with joints and body movements, we're going to look at joints. And I see that my chat is going off. Sorry.
I have a question.
Yes, what's up?
Okay, so for the hinge, what's the example again? I'm sorry.
So the hinge joint is like any joint that kind of opens and closes. So if you think about it, have you ever heard of like a door hinge, like a hinge on a door? What that is like this structure thing right here, that's like the hinge of a door. And so you can open the door or close the door. And that hinge goes like that and like that. Same with like the knee. The knee is my favorite example of a hinge. It can open and it can close. Open, close. So a hinge is anything that involves flexion and extension. So a hinge joint is anything that like opens and closes like a hinge of a door.
Okay, and what was pivot?
Pivot is whenever one of the bones stays stationary or still while the other bone rotates around it. So like in basketball, if you like, I don't know all the rules, like if you like put 2 feet down while having like the ball in your hand, you can only pivot, like you can't like take another step. You have to leave 1 foot down and then you can kind of like rotate the other foot around to aim. So the example I gave was the radio-ulnar joint. The radius and the ulna, the joint formed in between the two. Whenever you do that, the ulna stays still while the radius pivots around the ulna. So the ulna is stationary and the radius pivots around the ulna. So any bone that like stays still while another bone pivots around it. And the radio-ulnar joint is the only pivot joint that I know of. So that's the only one you would like see.
Okay, thank you.
For sure. Okay, so those were all the joints we're gonna look at, and we'll quickly review our movements. So we looked at these at the beginning of the semester, so you should kind of know these. If you don't know them by now, you definitely want to drill these down. ESS professors, especially the level 3, level 4 professors, will say things like pronation, supination, inversion, eversion real quick in the middle of a sentence, and if you don't know it, you're gonna be really lost. Like for example, in biomechanics, the first week he was already talking about flexion and extension. We were looking at a muscle in the arm, and he goes, whenever you flex it, it does this. Whenever you extend it, it does that. And for me, I was like, crap, I don't really remember flexion and extension. So you want to get these down now. It'll make the rest of your college career so much easier. If you do not get these down, you will be so stressed in your upper level ESS classes. So get these down. So angular movements is just looking at a group of movements. We're not going to focus too much on that. We're going to look at these specific movements. So first we have listed here circumduction. That's just making a circle. Anything that can like go in a circle. So circumduction, anything that can kind of go like that. Extension, hyperextension, and flexion. So extension is whenever it's at a 180-degree angle. Flexion is whenever you decrease the angle. So we're looking at the elbow joint here. It's 180 degrees. When we decrease that angle, that's flexion. If I was double jointed and could get my arm to go even further up, that would be hyperextension. I can give another example. When you look at the hips, this is extension, or well, I guess we'll look at the knee because the hip joint's a little more complicated. So this is extension because that knee joint is at 180 degrees. Flexion because we decrease the angle. So extension, flexion, and then hyperextension would be if I could go even further than that, like if I was double jointed in my knee. Of the wrist, this would be extension, flexion, hyperextension. Extension, flexion, hyperextension. Oh, this is so hard. See, flexion and extension messed me up. Okay, flexion, or sorry, no. Oh gosh. Okay, extension, flexion, hyperextension. Extension, flexion, hyperextension. So yeah, I normally don't put flexion and extension on unless it's an easier example because for some reason that does not click in my mind. Then we have gliding, which you know is just movement of smooth surfaces. Not really too concerned about that. Rotation, you know, not super concerned about that. Supination and pronation. If you remember, supination is palms up. So like you're holding a cup of soup, or you're saying what's up. And then pronation is palms down. So supination, you're holding a bowl of soup, or you're saying what's up, supination. Pronation, palms down. Inversion, eversion, dorsiflexion, and plantar flexion are all looking at the foot. So this is inversion, your ankle is coming in, and then eversion is your ankle going out. Inversion, eversion, inversion, eversion. Then we have dorsiflexion and plantar flexion. Plantar flexion is planting your foot down. You're planting your foot down. Dorsiflexion is bringing your foot up. So plantar flexion, dorsiflexion. Plantar flexion, dorsiflexion. And then we also have abduction and adduction. So this is abduction, that is abduction, adduction, or adduction, or adduction, adduction. So anything that's being taken away from midline, you're being abducted. So abduction, away from midline. Adduction, you're adding to midline. So abduction, you can think abduct, you were abducted, you were taken away. Adduction, you're adding to midline, so coming closer. Then we have depression and elevation. When we're looking at elevation and depression, and also protraction and retraction, we're only looking at the jaw and the shoulder blades. So with elevation, with the shoulder blades, elevation, depression, when you're looking at the jaw, elevation is depression is elevation, depression, elevation, depression. When we're looking at protraction and retraction of the scapula, protraction is this, retraction is that, protraction, retraction, same thing with the jaw. Protraction is, and retraction is protraction, retraction. So that's looking at those two. So I drank a giant Diet Coke before this, and I have to pee really bad. So y'all give me 5 minutes, and y'all can do these activities on your own to kind of identify those joints that we just went over. So I will be back in literally like 2 minutes. Feel free to do this and pull up the next PowerPoint while I'm gone. Sorry, there's no bathroom on this floor, so I have to climb 2 floors. Okay, so hope you have fun with that. I'm back. So that was it for our joints and body movements, and now we are going to get into our muscular system. So as ESS majors, I'm sure a lot of you have interest in muscles and exercise, things like that. Today we're going to be looking at the histology aspect, not the, oops, sorry. Oops, did not mean to say that. I closed the PowerPoint on accident. So today we're not going to be looking at the specific functions of the muscles or where they're located. We're actually just going to be looking at the muscle cells today. From what I heard from my Tuesday's lab is that y'all have not started muscular system in lecture yet. So this is probably going to stress a lot of you out. There's a lot of big new words that we're going to learn today. Whenever you learn it in lecture, it'll kind of get pulled together. I worked on a new diagram that can hopefully make it make a little more sense, but yes, you might leave this lecture a little frustrated and confused. If that happens, definitely feel free to email me or anything and we can get it solved, but hopefully I can get it across somewhat to where you can understand. So let me share my screen and put it in full screen. So this is chapter 10, getting into skeletal muscles, the histology and muscle anatomy. So what we're going to learn today, you have to know this for the rest of your career. Exercise physiology, resistance training and conditioning, biomechanics, all look at this. So you do need to know this. And if you do know it now, whenever you get to XPhys, you will do so much better. Just FYI, if you are health and fitness management or clinical exercise health and exercise physiology will be the hardest class you have to take. I took it this summer. It was very hard. I spent about 10 to 12 hours a day studying for 4 weeks straight in summer. Thank God it was like a pandemic and I didn't get FOMO that bad. I still got FOMO because I was sitting in my apartment and I was at another apartment and the study rooms were closed and my cats would always distract me. So I literally had to move all the clothes out of my closet and put my desk inside my closet and shut the door and put a lamp in there. And that's how I studied, was in a dark closet for 12 hours a day. But if you get your anatomy down now, XPhys will be a lot easier. I did make a really good grade in the lab. A lot of people have to, or in the class, a lot of people have to take it 2-3 times. So if you have a good base in anatomy, you won't have to take it a lot of times. So make sure you have a good base and you're kind of getting what I'm telling you today. So when you get to those harder courses, you can succeed in them too. So let's get into it. So our objectives are going to be looking at the different types of muscle tissue. We're going to look at the structure and function of sarcomeres. We're going to look at the components of a neuromuscular junction. And then we're going to look at ATP, which we actually aren't going to be looking at ATP. And we aren't going to be looking that much detail at the neuromuscular junction. I'll save both of these for you to learn in lecture, but these two we will definitely be learning. So if you remember, whenever we had our lab over tissues, the second week, we learned that there are 4 major tissue types: epithelial tissue, which was like our simple squamous, our pseudostratified, ciliated, columnar epithelial tissue, my favorite one to say because you sound super smart. We have like, you know, stratified, cuboidal, things like that. Then we had connective tissue, which was looking at like areolar tissue, adipose tissue, reticular, the blood, the compact bone. Those were our connective tissues. Now we're to our muscular tissues. So we have 3 main types of muscle tissue: smooth muscular tissue, skeletal muscular tissue, and cardiac muscular tissue. Now this is not written down anywhere on the slide, so you're going to want to make sure to note this. Smooth muscle, a lot of you probably haven't heard of smooth muscle. You've probably heard of skeletal muscle and cardiac muscle. Smooth muscle is the muscles that line our GI tract. So they line, you know, our esophagus, they line our stomach, they line our intestines, they line the genitalia, both male and female genitalia, and the rectum and the anus. So what smooth muscle is it's a muscle that provides very soft contractions. Compared to skeletal muscle, like our biceps and our triceps, those will provide very large contractions. So, you know, you can push a big barbell, you can, you know, pick up a baby, you can do larger contractions with skeletal and cardiac compared to smooth, because think about it, if you eat something, you don't want these giant contractions in your GI tract. I mean, we would all have to wear adult diapers if that was the case. We want smooth wave-like contractions, and that's actually termed peristalsis, but don't worry about that yet. We'll learn that in the digestive. But what it is there's this contraction called peristalsis that occurs in the GI tract. If you don't know what the GI tract is, it's the gastrointestinal tract, so all the way from your mouth all the way down to your anus. So the tract or the tube, that food comes in and then comes out. And that's where smooth muscle is found. It's also found in, some other locations, like I think it might be found in the sinuses. It lines our blood vessels and our nerves, but the main thing you hear when talking about smooth muscle is the GI tract, the gastrointestinal tract. And we have these smooth wave-like contractions. Like if you think in the stomach, like your stomach isn't like, get all the food out of there. It actually provides these kind of wave-like contraction, or these churning contractions, real soft. So smooth is the GI tract. Skeletal muscle is like any muscle that you voluntarily control. So like the muscles of my jaw, I'm talking right now. The muscles in my hands, my fingers, all that, everything I'm moving. It's not like my body is doing that on its own. Like I have to kind of control that. Even though sometimes, when I'm talking and do things with my hands, I'm not necessarily thinking like, do that with your hands, but I am controlling that. If I was thinking, okay, be still, like I'm controlling being still. So skeletal muscle is anywhere that like you have voluntary control. And then cardiac muscle is found in our heart. So let's kind of break down what differentiates these types of muscle tissue. So this is a really important chart, and I made the chart up. You might see it in just like a general format, but I put it into a chart just so it's a little bit easier to remember. And the two things we're looking at here are structure and control. Those are the two things that differentiate the three different muscle tissue types. So first we'll look at the structure. So when we're looking at the structure, we're looking at these things called striations. Striations are lines on a muscle that provide for stronger muscle contraction. And we'll look specifically at what striations are in a couple slides, but as of now, just think of them as muscles that provide for stronger muscle contraction. So smooth muscle is non-striated because again, we don't want those strong contractions. Let's say you just ate some like Torchies or Taco Bell or something. You don't want a strong contraction of that. It would just go straight through you and that would not be fun. It would stink. It would be a disaster. So we want just these smooth contractions. And it's not like you just eat food and it just gravity pushes it down. Once it gets to the stomach, it's got to go through the small intestines, which do something like that, and they all kind of, go around each other. So yes, there is muscles that will get that through us, but we want it to be slowly and smooth. So smooth muscle, non-striated, do not want strong muscle contractions. Skeletal muscle is striated. We do want strong muscle contractions. So we want to be able to, flex our muscles really hard. We want to be able to load a lot of weight. We want to be able to hold heavy things. We want to be able to push things, pull things, all of that. So we want those strong muscle contractions in our skeletal tissue. And then the cardiac tissue, the heart does have its own muscle tissue. When we get to the cardiovascular system, we'll learn it's actually called the myocardium, the muscle of the heart. That's what it means in medical terminology. You don't need to know that now. But cardiac tissue, or the muscle tissue of the heart, is also striated, because think about it. Your heart is this, fist-sized organ inside your chest right here, and it has to pump blood. This little tiny muscle has to pump blood all the way against gravity to the top of our heads, all the way to our fingers, all the way to our toes. I'm 6 foot tall. That's a really long way to get to. So we need striations of our cardiac tissue so it can have a strong pump, a strong contraction to get that blood all the way down to the toes, all the way to the top of the head, all the way to our fingers. So smooth is non-striated, don't want those strong contractions. Skeletal and cardiac striated, so we can have those strong contractions.
Did you say you were 6 foot?
I am 6 foot tall. Yes, I am very tall.
Wow. Okay.
I know, teaching y'all on Zoom, y'all probably don't notice that about me, but I'm very tall. So if y'all ever see me on campus, y'all might be like, so yeah. But yeah, so now we'll look at control. So smooth muscle is involuntary. So you don't eat something and then think, okay, go down my throat. Okay, go to my stomach. Okay, go to my small intestines. Okay, go to my rectum. Go to my hands. You don't think that. It just, it's involuntary. It does it on its own. Skeletal muscle, however, yeah, whenever you want to lift your arm up, whenever you want to do something with your skeletal muscles, whatever it may be, you are controlling that. You are making that happen. Cardiac muscle, no. You do not think, okay, beat, okay, beat, okay, beat, okay, beat. It does it on its own, or all day long we'd be sitting there thinking beat, beat, contract, contract, contract. That's impossible. We would not be able to, sustain life. So cardiac muscle, involuntary, beats on its own or contracts on its own. Skeletal muscle, voluntary, those are all the muscles that you think of whenever you think of muscles, so like biceps, triceps, the quads, the calf muscles, all of those are our skeletal muscles, voluntary. And then smooth muscle, involuntary. Whenever you eat something, you don't think, okay, go here, go there, go there. So definitely want to know this chart. So now we're going to go into the scary part. We're going to get into the structure of a whole muscle. So probably after tendon, you probably haven't heard any of these before. So yes, these are big and new scary words. I remember when I was in A&P and I saw these for the first time, I was like, what in the hell? How is this even possible to know this? But it really is not that bad. Repetition is key. Understanding this is key. Drawing this over and over again. And in our lab reports today, there will be a drawing assignment of this and of this. So repetition is key for this. So let's get into it. First of all, major mistake right here. Tendon is bone to muscle. Ligament is bone to bone. Major mistake in my slide. Please fix that. Tendon is bone to muscle. Ligament is bone to bone. So tendon, bone to muscle. Ligament, bone to bone. So epimysium, paramysium, and endomysium, I'm not concerned about those at all. Those are just coverings of the muscles. So I'm not even going to go over those because those are big words you don't need to stress about right now. So epimysium just covers the entire muscle. Paramysium covers an inner portion, and endomysium covers the muscle cell, but we're not going to stress about those. So we have our tendon that's connecting our bone to our muscle. And whenever we look at that muscle, there are a bunch of these bundles. And what they are bundles of muscle fibers. Note this down. Anytime you hear the word muscle fiber, I am referring to a muscle cell. So just like we have our normal cells, just like we have the cells of the nervous system, our brain cells. We also have muscle cells. We have blood cells, we have cardiac cells, we have a bunch of different type of cells. Whenever we're talking about a muscle cell, they call it a muscle fiber. I'm not sure why they call it a muscle fiber and not just a muscle cell, but anytime you hear a muscle cell, think muscle fiber. And just like regular cells, muscle fibers or muscle cells also have organelles. So like how we have the mitochondria, how we have the Golgi apparatus, all of those, so do muscle cells. They just have different weird names. So for example, we have the plasma membrane of a regular cell. The plasma membrane of a muscle cell is called the sarcolemma. So if you want to put in parentheses like plasma membrane, sarcolemma, same thing. Just like we have the cytoplasm of a regular cell, we have the sarcoplasm of a muscle cell, the cytoplasm of a muscle cell. We also have nuclei or nucleuses in our muscle cells. We do have mitochondria in our muscle cells too. If you remember, mitochondria is the powerhouse of the cells. So in our muscle cells, we have a bunch of those. And then we have a few other new types of organelles that you've probably never heard of before, like myofibrils, sarcomeres, sarcoplasmic reticulum, all these things. You're probably like, what the heck? For this lab, we're just going to focus on these two because they're the most important, and the sarcomere is very important. So inside of our muscle fiber or a muscle cell, we have a bunch of myofibrils. Myofibrils are organelles that are actually responsible for contraction. So they're what cause the contraction. And making up the myofibrils are these structures called sarcomeres. One myofibril is about 10,000 sarcomeres. So in one myofibril, we have about 10,000 sarcomeres. And this is just the definition part. If you're not a definition part, if you're more of a visual person, we're about to get there. I'm definitely more of a visual person. So if you're sitting here like, oh my gosh, I'm gonna give up. Don't yet. We're about to get to the visual, and then we'll get to a diagram that will hopefully, reinforce it in your mind. So one myofibril has about 10,000 sarcomeres, and yeah, that's the main parts of a muscle cell, the myofibrils and sarcomeres. Whenever you're in lecture, you'll learn what the sarcoplasmic reticulum is, what the terminal cisternae, the T-tubules, and the triads are. I'm not going to stress you all out about those now. You'll definitely look very closely at sarcoplasmic reticulum and T-tubules and exercise physiology. Very, very, very intensely look at those, but not yet. So maybe just kind of study them a little bit so when you get to X-Phys, you have an idea, but for now, you're good. So let's look at a visual of all this. So we have our tendon. You can pretend right here is like a bone. So this could be like the humerus, and now we're going to look at the biceps. So here's the bicep. Just pretend this is the bicep. I wish I had that. Let's see, imitate, draw. So here's our humerus, our arm bone, and then we have our biceps. With those striations because it's a muscle cell or a skeletal muscle cell. And so we're looking at the biceps. That's what's right here. Whenever we cut into the bicep, we have these structures called fascicles. So these guys are called fascicles. And what fascicles are bundles of muscle fibers or muscle cells. So this is still a fascicle. And then we get into this is like one muscle cell. So like just a regular cell. This is our muscle cell. And what you see inside of it are all those organelles. So we have the sarcolemma, the plasma membrane of the muscle cell. Sarcolemma, decides what gets in and out. We have, nuclei, and we have mitochondria in there. But most importantly, we have myofibrils. These are myofibrils. If you break the name apart, myo means muscle, and then you can think fiber. So make up a bunch of muscle cells. And inside our myofibril, we have about 10,000 sarcomeres. So this right here is one sarcomere. This is another sarcomere. This is another sarcomere. We have about 10,000 of those in one myofibril. So with my little spotlight, I'll take us through this one more time. Oops. Just kidding, it's not letting me draw. So we have our bone. Oh, stop it. My computer's messing up. So we have our bone. Okay. So we have our bone, like our humerus. We have our muscle, for example, our biceps. You look at the muscle right here. Inside the muscle, we have a bunch of bundles called fascicles. These fascicles contain a bunch of little muscle cells. So inside these fascicles are a bunch of muscle cells. When we look at the muscle cell as a whole, we can see that it has a lot of little organelles in there, most specifically the myofibrils. Inside each myofibril are about 10,000 sarcomeres. So that hopefully makes a little bit of sense to you.
Is the one that you circled in white, like the biggest thing, is that like one muscle cell?
So yes, oops. So this right here is one whole muscle cell. And a lot of the time muscle cells are drawn out in this weird like structure. They're not like a regular circle like you normally see with the nucleus in the middle and the mitochondria over here and the Golgi apparatus over here. The muscle cells are normally put in this weird kind of like oval-like shape, just because that's how most muscle cells are shaped. So yes, this entire thing right here is the muscle cell. Okay. Cool. Let me see the chat.
Okay, thank you.
How many myofibrils in a muscle cell from Colin? So that is actually determined on how fit you are. So if you resistance train a lot, how you get that appearance of bigger muscles, like for me, my bicep is pretty small. Some, people have like giant biceps that come like way up here. That's because they have worked out enough. They have developed more and more muscle cells. When you develop more and more muscle cells, you get more and more myofibrils, and those will enlarge the muscle cells, and that's what makes you get, bigger looking muscles. So there's no specific number of myofibrils in one muscle cell. It depends on how much you resistance and strength train. The more you strength train, the more myofibrils you'll have, and therefore the bigger your muscles will look. It took me a long time to learn all this. I did not drill this down until this summer when I took XPhys. It took a lot of practice when I was in lab. And so, yeah, it gets really stressful. Just FYI to all ESS majors.
I have a question.
Yes, what's up?
What are the bundles inside the fascicle called?
Those are called the muscle fibers, aka the muscle cells. So I can actually draw a little thing on the board. So once we look at the structure of the sarcomere next, I have a diagram that kind of pulls it all together. So whenever we're looking at a whole muscle structure, so we have the bone. We have the bone, we have the tendon, we have the muscle. So again, tendons connect bone to muscle, ligaments connect bone to bone. So the tendon connects the bone to the muscle. Whenever we dive, so now we're inside of the muscles, I'll change colors. So inside the muscle, we have our fascicles. Inside the fascicles, we have our muscle fibers, aka our muscle cells. I hope y'all can see that. Inside the muscle fibers or muscle cells, we have myofibrils, and inside myofibrils, we have about 10,000 sarcomeres. Per one myofibril. So maybe draw that down kind of in a chart format. So we have our bone, Connecting the bone to the muscle is the tendon. So we get into our muscle, we look inside the muscle. We have our fascicles, bundles of muscle cells or muscle fibers. When we actually look in the muscle fiber, the muscle cell itself, we have myofibrils, which are just an organelle, just like a mitochondria, just like a nuclei, just like a lysosome, all that. We have a myofibril, which is an organelle. And inside one myofibril, we have 10,000 sarcomeres. Hopefully that kind of made it make a little more sense. So that's the structure of a whole muscle. Now we're going to look at the structure of a sarcomere. So our last step, our sarcomere. So we're now inside the sarcomere. So sarcomeres are what give skeletal muscles that striated appearance, those lines. Those lines are caused by the appearance of sarcomeres. They're also what cause stronger muscle contractions. So sarcomeres are what control our contractions. In a contracted muscle, they're shortened or narrowed, and in a relaxed muscle, they're stretched out. So whenever you flex your muscle, what is happening is the muscle is actually narrowing or shortening. So it's narrowed. Whenever I relax my muscle, it lengthens. So whenever you contract a muscle, it shortens. Whenever you relax it, it lengthens. So in a contracted muscle or a flexed muscle, sarcomeres are narrowed. Whenever we relax our muscle, they lengthen. And we'll look at how that happens in just a second. So that is all based off of these two proteins called actin and myosin. Actin and myosin. And these are little filaments that will attach to each other and cause that narrowing and lengthening. And this is easier to explain visually. So this one's going to be kind of harder for me to explain definition-wise. So let's just jump straight into the visual. So here is our sarcomere. Again, we have 10,000 of these in one myofibril. So 10,000 of these guys right here in one myofibril. You look inside, and those two protein filaments, actin and myosin, are these guys right here. Actin is the thin filament, so these are actin. Myosin is the thick filament. These are myosin. A way I remember that is like myosin, myo, the prefix myo means muscle. So you can think like, oh, I want thick muscles, like you want thick, big muscles. So myosin is that thick filament. Actin, you can think like actors always say that the camera adds 10 pounds, so they always want to be thin. So actin, thin, myosin, thick. And anchoring these two filaments are these lines called the Z line right here and the M line right here. The M line is the middle of the sarcomere. You can see it right there. That's the M line. And the Z line is where the actin anchors. And it's also where each sarcomere kind of like becomes its own. And I'm going to draw this in a diagram that'll help. We also have the A band and the I band. The I band is right here. If you look at a muscle, there are portions that look lighter, there are portions that look darker. That's why it kind of looks like it has lines. The I band is just thin filaments, so I band is just actin. And the A band, which is this whole thing right here, is thick and thin filaments, actin and myosin. So it's the darker band. The I band is the lighter band. I band, lighter band. A band, darker band. So let's draw this out. So I'm actually using a diagram from exercise physiology. I've definitely made it a lot more simple, but and I've added a little bit of my own to it. So let me do some erasing real quick. I think I have a picture of this on my phone so I don't forget it. I made this diagram up while y'all were taking your quiz so I don't want to lose it. Alrighty, okay. So you definitely would probably want to write this down on a piece of paper right now if you have access to one. So really, can y'all see what I write? Is it like really hard to see, or is it like kind of easy, like not too bad? Anyone? I forget that for some reason my chat always disappears when I switch from slide to slide. Yeah, so if you're following along, definitely want to like make your screen and like full screen right now. So we're gonna start from the whole muscle. So we have our person. So here's our dude, and they are showing off their strength. So here is their bicep. Y'all are gonna have to like unmute yourself if you can't like see something, because I can't tell from my little screen. So here's our dude, here's his bicep. So we're gonna enlarge this. So here is his humerus, and here is that muscle on top of it. Here's the muscle, and then a tendon that's attaching to. Sorry, the motion sensor went off. So we look inside this muscle, and comprising the muscle are the fascicles. So remember fascicles. I guess I should color code this. Go from there. So we're looking at the biceps or any skeletal muscle, and we have the fascicles. And inside the fascicles, remember we have a bunch of muscle cells, or muscle fibers, or what they're technically called. So here is our muscle fiber. And inside the muscle fiber, we have myofibrils. We also have nuclei and mitochondria and all that, but we're looking at the myofibrils right now. Inside the myofibrils, we have, so actually I'm going to show y'all what one myofibril looks like. So we're going to look, here is one myofibril. And one myofibril, we have about 10,000 sarcomeres. So here's our myofibril, here's our sarcomere. Let's enlarge the sarcomere. So when we enlarge the sarcomere, it looks something like this. This is one sarcomere. Here's another sarcomere. So this is a sarcomere, 1, 2. Inside the sarcomeres, we have thick and thin filaments. Attaching, these are right here, are called the Z lines. They kind of look like Z lines. The Z lines are what separate each sarcomere from each other. So sarcomere, Z line, another sarcomere, Z line, another sarcomere, Z line, so on and so on. Attached to the Z lines are our thin filaments, actin. So here are our thin filaments, actin. And so on. Then we have our thick filaments, which are called myosin. Myosins are in the middle. Middle. And these are thick filaments. Each myosin filament has a bunch of things called myosin heads. So they have these heads that will attach to actin. And whenever you want to flex or contract your muscle, those myosin heads will pull the actin filaments closer to the end line. Here is the end line. So whenever we want to contract our muscle, those myosin filaments, the thick filaments, will attach to actin, the thin filament, and draw them into the end line. And that's why whenever you contract a muscle, it shortens. Whenever you relax it, it lengthens. Whenever you relax it, those heads let go of the actin, and it lengthens, relaxes. Then when we look at the A bands and the I bands, the A band is just where the thin filaments are. So just right here, that is the I band, the lighter band. So right, I band, only thin filaments. I band is only thin filaments. And then we have the A band, which is thick and thin filaments. So the A band, thick and thin filaments. Also known as the darker band. So you can think here. I band is the lighter. Hang on, I know y'all probably can't see that. And then the A band is the darker. So you can kind of use that as like a memory, like I band is the lighter band, A band is the darker band, because it contains actin and myosin. And the I band contains only actin. Okay? So, probably very scary for y'all. I remember those days. I was very stressed. But you can see that I can voice it to y'all now. I'm not like a super smart genius at all. I just have learned to study and how to get yourself motivated for things. So this is doable if you put in the work. You can understand this, and you can understand it to a point where it becomes interesting. So I'm going to go over it one more time with this picture. We have our thin filaments, which are called, and you don't have to answer on the mic, I'll just kind of pause for a second so you can recall it in your head, kind of forces you to relearn it. So we have our thin filaments, which are known as actin. We have our thick filaments, which are known as myosin. The actin attaches to the Z line, looks like a Z. The myosin attaches to the M line, middle line, the middle of the sarcomere. We have the little myosin heads that attach to the actin and bring those Z lines together. And we have two bands, the I band and the A band. The I band contains only thin filaments, and the A band contains thick and thin filaments. The I band is the lighter band and the A band is the darker band. Lighter, darker. So this is your sarcomere, and the sarcomeres are how we contract our muscles. Exercise. Oh, go on.
What would you quiz us on about this part? Like, what would you ask us?
So I would either like post this picture right here, and I'd have something like blanked out, and I'd say, what is this structure identifying? Or I'd say, blank is the thin filament, or the myosin attaches to which line. So like everything on the sarcomere is fair game. So I could say like the blank band is the darker band, or I could say the blank band contains myosin and actin, or the A band contains blank, or the I band contains blank. So everything here is fair game. Definitely be able to identify the picture, you're not responsible for H zone, but everything that's boxed in, be able to identify. Anything that, I left blank here, be able to identify. And yeah, the labeling and kind of just the, general idea of it. So yeah.
Okay, thank you.
So real quick, we're gonna watch this video and then we'll be done we have one more slide, but I'm gonna go through it like in 30 seconds. But we're gonna watch this video to kind of pull it all together. This is the sliding filament theory. This is what you'll actually be learning in lecture. This is a very complex theory. It's about muscle contraction. At Texas State, they call it the sliding filament theory. I always heard it as just called, the theory of muscle contraction or muscle contraction physiology. So we're gonna watch just a little clip. We're not gonna watch this whole video, but we're gonna watch what we just went over so it kind of will make sense to y'all. So let me see if it'll play. If not, I'm gonna have to go to YouTube to get it to go on because for some reason it's worked every semester for me until this semester. And open movie in browser. Sharing is paused. Hey. Here we go. It hasn't. I watched it on Tuesday. Okay, just a second here. Wi-Fi this. Okay. What was it called? The Okay. All right, so we're just gonna watch it first. Yeah, calming videos for cats. Every time I'm studying, that's like the only time my cats want to play with me, so I always play like interactive bird videos for them to watch. Okay, so just watch this. You don't need to take notes on it. Just sit here and watch it and see if it kind of ties everything together for y'all.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei.
So real quick, here's our muscle. This is muscle, then this is a fascicle that contains a bunch of muscle fibers.
Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound...
So yeah, after that point is when you're going to learn about ATP and all of that. You'll learn that in lecture. So yeah, if you think this is complicated, it's going to get a lot more complicated in lectures. So let me return to our PowerPoint. We're almost done for the day. I know, I'm sorry, it's been a long lecture, but you know, got to get through it. So just for my lab, the general thing I want you to know for sliding filament theory are that those Z lines are pulled towards each other as actin slides over the myosin. Well, sliding filament theory, the Z lines are pulled towards each other due to actin and myosin. So we're not going to go over neuromuscular junctions or motor units much at all. Nerve cells will communicate with the plasma membrane of a muscle fiber and send a neurotransmitter such as acetylcholine or ATP to the muscle cell to cause it to contract. When you look at a motor unit, it's just a neuron, which is a nervous cell, innervating a fiber. I'll save that all for nervous system. So don't worry about this, you'll learn in the lecture too. So tropomyosin and troponin, definitely don't need to know that for lab. You'll learn that in lecture, and you'll definitely learn it in ex-phys, but for lab, definitely not. So, but good question. I'm glad you know those words. So that is it for today. Let me go over the lab report really quick before y'all leave. Oh my gosh, my computer's gonna die. See that. Okay, so our lab report today, go back to Canvas. Where is Canvas? Canvas, what did you call? Close that out. Here we go. Zoom, where did Zoom go? Close, operating back to Cool. So if you go to today's module, you can find today's lab report, which is lab report 5. And what I'm going to have you do is, of course, attach your outline, and then you're going to draw the sarcomere and label the following structures inside. So label all of those. Here's like kind of just an example. This is actually from my exercise physiology class this summer, one of the diagrams he gave us to draw out. You definitely don't need to know about the H zone, shortening, then lengthening, or anything like that. But definitely know like the Z disc, the actin, the myosin, inline, the Z line, like all of that, and draw it out. I did not get to show my Tuesday lab, the part where I gave you all the diagram of the muscle and then the muscle fiber. So I might draw that out in just a second and attach it to this assignment, and you can draw that too. I will accept just this, but if you want to, for your own learning, to add the part that I added today, it will only benefit you in the long run. And it's just a drawing. It's not like it's hard. I actually hope y'all are enjoying these assignments. I think, you know, like fun work is way better than just like reading a book and like writing an essay over it, which is what most of the other IAs do, is just have y'all write like a 500-word essay each lab. And I'm like, that sounds not fun and like a bullshit assignment. So hopefully you'll enjoy these. If you don't, email me and I'll set something up to where you can just write an essay, but I think these are fun. So again, I ask that y'all attach this in one Word document. So do your outline and then take a picture and then put that picture into the Word document. So whenever I download all of the submissions, it doesn't like break my computer and make it super slow. I don't want to have to go to campus all the time to grade things. So please just put it in a Word document, attach it there. And that's it. So thank y'all so much for staying. I'll hang around for extra questions if y'all have anything. If not, be prepared for a long week next week and you don't want to miss it. Also, yeah, if you have a grading issue, go ahead and either email me or chat me right now. Okay, cool. Olivia, I have that noted. By Colin, Olivia, I will get that taken care of right away. Thank y'all.
Bye.
Bye. Righty, y'all got questions for me? No. Can you check our chat? Oh yeah, Jaden, I do have a message that you chatted me on there. I will for sure go and check that out. I think the issue that I was having was like I couldn't see like your drawings or something, but I will go look at that right away. Okay, cool. Alrighty, have a good weekend. Bye.